Hey there, amazing people. I'm thrilled to have you here today. Hello and a warm welcome to our channel. Today, we're delving into acidophiles in acid mine drainage. The outflow of acidic liquids and other pollutants from mines is often catalyzed by acid-loving microorganisms. These are the acidophiles in acid mine drainage. Acidophiles are not just present in exotic environments such as Yellowstone National Park or deep-sea hydrothermal vents. Genera such as Acidithiobacillus and Leptospirillum bacteria and Thermoplasmatales archaea are present in syntrophic relationships in the more mundane environments of concrete sewer pipes and implicated in the heavy metal-containing sulfurous waters of rivers such as the Rheidol. Such microorganisms are responsible for the phenomenon of acid mine drainage (AMD) and thus are important both economically and from a conservation perspective. Control of these acidophiles and their harnessing for industrial biotechnology shows their effect need not be entirely negative. The use of acidophilic organisms in mining is a new technique for extracting trace metals through bioleaching and offers solutions for acid mine drainage in mining spoils. As we venture forward, let's examine the introduction in detail and gain a deeper appreciation for its significance. Upon exposure to oxygen O and water Ho, metal sulfides undergo oxidation to produce metal-rich acidic effluent. If the F is low enough to overcome the natural buffering capacity of the surrounding rock's calcium carbonate equivalent or acid neutralizing capacity, the surrounding area may become acidic, as well as contaminated with high levels of heavy metals. Though acidophiles have an important place in the iron and sulfur biochemical cycles, strongly acidic environments are overwhelmingly anthropogenic in cause, primarily created at the cessation of mining operations where sulfide minerals, such as pyrite iron disulfide or Fis, are present. Acid mine drainage may occur in the mine itself, the spoil heap, particularly colliery spoils from coal mining, or through some other activity that exposes metal sulfides at a high concentration, such as at major construction sites. Banks et al. Provide a basic summary of the processes that occur. As we progress, let's zoom in on bacterial influences on acid mine drainage and examine its role in shaping our overall narrative. The oxidation of metal sulfide by oxygen is slow without colonization by acidophiles, particularly Acidithiobacillus ferrucens, synonym Bacillus ferrucens. These bacteria can accelerate pyritic oxidation by times. In that study, a proposal for the rate at which a ferrucens can oxidize pyrite is the ability to use ferrous iron to generate a ferric iron catalyst. Fe plus EO plus HV plus Tho. Under the above acidic conditions, ferric iron Fe is a more potent oxidant than oxygen, resulting in faster pyrite oxidation rates. A ferrucens is a chemolithotrophic bacteria due to the oligotrophic nature, low dissolved organic carbon concentration of acidic environments, and the lack of illumination for phototrophy. Even when in those conditions, a ferrucens can survive if the rock retains moisture and the mine is aerated. In fact, in this situation, with pioneer microorganisms, the limiting factor is likely to be the environmental circumneutral, which inhibits many acidophiles' growth. However, favorable geochemical conditions quickly develop with an acidic interface between the bacteria and the mineral surface, and F is lowered to a level closer to acidophilic optimum. The process proceeds through a ferrucens exhibiting a quorum level for the trigger of acid mine drainage (AMD). At first colonization of metal sulfides there is no AMD, and as the bacteria grow into microcolonies, AMD remains absent. Then at a certain colony size, the population begins to produce a measurable change in water chemistry, and AMD escalates. This means F is not a clear measure of a mine's liability to AMD. Culturing a ferrucens or others gives a definite indication of a future AMD issue. Other bacteria also implicated in AMD include Leptospirillum ferrucens, Acidithiobacillus thusans, and Sulfobacillus thermosulfideucillans. As we venture forward, let's examine archaean acidophiles in detail and gain a deeper appreciation for its significance. Though Pseudomonadota, formerly Protobacteria, display impressive acid tolerance, most retain a circumneutral cytoplasm to avoid denaturation of their acid-labile cell constituents. Archaea such as Ferroplasma acidophyllum, which oxidize ferrous iron, have a number of intracellular enzymes with an optimum similar to that of their external acidic environment. This may explain their ability to survive as low as 1.3. The differing cell membranes in Archaea compared to the bacteria may hold part of the explanation. Ether lipids that link isoprene, compared to Pseudomonadota's diester linkage, are central to the difference. Though lacking a cell wall, Fahrenheit. Acidophyllum cell membranes contain caldarchated glycerol tetrahydrolipids, which effectively block almost all proton access. Thermoplasma acidophyllum also uses these bulky isoprenid cores in its phospholipid bilayer. It is possible that the family Ferroplasmataceae may in fact be more important in AMD than the current paradigm, Acidithiobacillus. From a practical viewpoint, this changes little, as despite the myriad physiological differences between Archaea and bacteria, treatments would remain the same if is kept high, and water and oxygen are prohibited from the pyrite. The reaction will be negligible. The isolation from sulfur-taric soils of two Picrophilus species of Archaea poschema and Torridus are of note for their record low survival at 0, indicating that further AMD microorganisms may remain to be found which operate at an even lower F. Though the genus Picrophilus is not known to be involved in AMD, its extreme acidophilia is of interest, for instance its proton-resistant liposomes, which could be present in AMD acidophils. With the groundwork laid, let's now examine interactions in the mine community and its connections to our previous discussions. Tentatively, there may be examples of syntrophy between acidophilic species, and even cross-domain cooperation between Archaea and bacteria. One mutualistic example is the rotation of iron between species. Ferrous oxidizing chemolithotropes use iron as an electron donor, then ferric reducing heterotrophs use iron as an electron acceptor. Another more synergistic behavior is the faster oxidation of ferrous iron when aferusidins and sulfobacillus thermosulfideuxidins are combined in loco-culture. Thermosulfideuxidins is a more efficient iron oxidizer, but this is usually inhibited by loco-uptake. Aferusidins has a higher affinity for the gas, but a lower iron oxidation speed, and so can supply thermosulfideuxidins for mutual benefit. The community possesses diversity beyond the bacteria and Archaea. However, the approximately constant present during acid mine drainage make for a reasonably stable environment, with a community that spans a number of trophic levels, and includes obligately acidophilic eukaryotes such as fungi, yeasts, algae and protozoa. Let's now turn our attention to physiology and biochemistry and uncover the fascinating insights it brings to the table. Acidophils display a great range of adaptations to not just tolerating, but thriving in an extreme environment. The definition of an acidophile being an organism that has a optimum below 3. Principle in these is the necessity of maintaining a large gradient to ensure a circumneutral cytoplasm normally, however not in Picrophilus species. The Archaeans have already been discussed above, and further information on their antibacterial adaptations are in basic form in the figure. To elaborate upon the figure, the bacteria also use membrane proton blocking to maintain high cytoplasmic F, which is a passive system as even non-respiring aferusidins exhibit it. Acidophils are also able to extrude protons against the F gradient with unique transport proteins, a process more difficult for moderate and hyperthermophils. A higher temperature causes cell membranes to become more permeable to protons, necessarily leading to increased H influx, in the absence of other membrane alterations. In this section, we'll be deep diving into proton motive force, unraveling its complexities and uncovering valuable insights. To grow at low F, acidophils must maintain a F gradient of several F units across the cellular membrane. Acidophils harness the strong proton motive force PMF caused by the F gradient across their cell membrane for ATP production. A large amount of energy is available to the acidophile through proton movement across the membrane, but with it comes a cytoplasmic acidity. Instead, ions such as sodium can be used as a substitute energy transducer. To avoid this increase, APAs are often null-linked rather than H-linked. In this part of the video, we'll be delving deeper into expelling H-containing vesicles and analyzing its key components. Expelling H-containing vesicles. Alternatively, bacteria can use H-containing vesicles to avoid cytoplasmic acidity, see figure, but most require that any H taken in must be extruded after use in the electron transport chain etc. On the subject of the etc., an adaptation to living in the mine environment is in the use of different etc electron acceptors to neutralophiles, sulfur, arsenic, selenium, uranium, iron, and manganese in solid form rather than most commonly Fe in dissimilatory ion reduction, frequent in AMD. In this chapter, we'll be unraveling the enigma of genomic adaptations and discovering its transformative power. Genomic adaptations are also present, but not without complications, in organisms like Thermoplasmatales archaea, which is both acidophilic and thermophilic. For instance, this order expresses an increased concentration of purine-containing codons for heat stability, whilst increasing pyrimidine codons in long open reading frames for protection from acid stress. More generally, and presumably to reduce the chances of an acid hydrolysis mutation, all obligate hyperacidophiles have truncated genomes when compared to neutralophile microorganisms. Picrophilus torridus, for instance, has the highest coding density of any non-parasitic aerobic microorganism living on organic substrates. Moving forward, we'll be taking a closer look at improved repair. Acidophiles also benefit from improved DNA and protein repair systems such as chaperones involved in protein refolding. The torridus genome just mentioned contains a large numbers of genes concerned with repair proteins. As we venture forward, let's examine biotechnology applications in detail and gain a deeper appreciation for its significance. I-mediation is the primary biotech issue created by the AMD acidophiles. There are a number of methods for dealing with AMD, some crude such as raising through liming, removing water, binding iron with organic wastes, and some less so application of bactericides, by control with other bacteria archaea, offset wetland creation, use of metal immobilizing bacteria, galvanic suppression. A number of other neutralizing agents are available: pulverized fuel, ash-based grouts, cattle manure, whey, brewer's yeast, many which solve a waste disposal problem from another industry. As supplies of some metals dwindle, other methods of extraction are being explored, including the use of acidophiles in a process known as bioleaching. Though slower than conventional methods, the microorganisms, which can also include fungi, enable the exploitation of extremely low-grade ores with minimum expense. Projects include nickel extraction with aphaerousins and Aspidillus sp. Fungi and sulfur removal from coal with Acidithiobacillus sp. The extraction can occur at the mine site, from wastewater streams, or the main weight occurs if the contamination has reached that far, in bioreactors, or at a power station, for instance, to remove sulfur from coal before combustion to avoid sulfuric acid rain. With the groundwork laid, let's now examine future of the technique and its connections to our previous discussions. AMD continues to be important in the river Rheidol, and in the near future further treatment will be needed in the area around Aberystwyth, which contains 38 of the 50 worst polluting metal mines in Wales. In 2007, the UK government endorsed a return to coal as an energy source, and mining in the UK is increasing. For instance, the open-cast coal pit at Fowes Wyfran, Merthyr Much preventative work will be required to avoid the AMD associated with the last generation of coal mines. The fast and efficient protein and DNA repair systems show promise for human medical uses, particularly with regard to cancer and aging. However, further research is required to determine whether these systems really are qualitatively different, and how that can be applied from microorganisms to humans. As discussed above, acidophiles can have the option to use electron acceptors other than oxygen. Johnson 1998 points out that facultative anaerobism of acidophiles, previously dismissed, could have major implications for AMD control. Further research is needed to determine how far current methods to block oxygen will working, in light of the fact that the reaction may be able to continue anaerobically. Stay connected and join our community by subscribing and following me on other platforms.